Hi, my name is Logan. I'm a master's intern here at PNNL. And so my time here, I've been working on this NEATO app. So this is going on the map framework that Rachel had talked about earlier. So basically currently in MAP, there's PMART, which does a great job at performing differential abundance analysis. But still, even after doing differential abundance, there can still be a lot of data and a lot of questions about like actually how the system is changing. So that's kind of when people would want to move into functional enrichment. So So basically, we wanted to add an app to the map framework that could perform the functional enrichment for users. So yeah, a big part of functional enrichment is networks, which do a great job of visualizing the results of functional enrichment and looking at the pathways and cell functions that are actually changing. So yeah, so basically this app focuses on generating networks and functional enrichment tables for identifying these systems that are changing. So here we can kind of see like these outer pictures are screenshots from the app, and then in the upper right corner, that's like kind of behind the scenes like architecture of the app. So kind of walking through how a user would use it, they upload their data, they specify some columns with the necessary data in it, they specify the species that they're looking at, and they specify some like parameters for filtering the data. And yeah, so then they would generate a network using one of the network algorithms that we've implemented. This network is interactive, so users can click and drag on a node to move it around. The width of the interaction is a measure of how confident the interaction database is in that interaction. So kind of like based off of like literature or experiments, they assign it a score, and that's represented in the graph here. And then that can be exported as a PNG as well. And then moving on to the table, that can be filtered by p-value or adjusted p-value. The table will actually update if you click on a cluster in the network. And so you can perform enrichment on just that cluster. And for the background of the enrichment, you can either use the full data set that you uploaded or just the network. And kind of moving on to the architecture, you'll kind of notice that there's lots of different algorithms, and that's because different algorithms have different pros and cons. So for example, in this picture, this was generated using the PCSF algorithm. That R package was kind of made here at PNNL. And what it does is it assigns a prize for every node, in this case, the log fold change, and the cost would be the confidence between the nodes. So basically, the algorithm just tries to find the best pathway through the node and develops this network, and that can help fill in some gaps that are missing in the data due to maybe the instrument not picking up certain proteins. It can give a more complete picture of the pathways in the cell being affected. Another thing that we want to do is you'll notice that there's a lot of different Docker containers kind of talking to each other, and it's because we want to make this app very modular. So we can exchange the protein interactions and the enrichment databases with those of different species. Right now, this app only supports human and mice, but part of the reason that we're here is that we want to talk to users and figure out, what species you would want to see supported and things like that. So yeah, I will be presenting at the poster session today. So yeah, if you're at all interested in this, please let me know and we can work that out. Yeah, thank you.
Thank you, Logan. Our next speaker is David Degnan, a data scientist here at PNNL. Please, David.
Hey there, can you hear me? Hey, hi, I'm David Degnan. I am part of the MAP team here at PNNL. Today, I'm going to be talking to you about the mode application, which Rachel talked about before, but it's part of helping us explore our omic data at many different steps here, in this case, following statistics like the output from PMART-R. So let's go ahead and start the video. To identify trends in an omics data set is a challenge. So for example, following analysis with PMARD-R, we can have 4 files that get generated. The first of which is the expression matrix, which this has been normalized. So each of the columns is a sample and each of the rows is a biomolecule. And with that, you're going to have information for each of your samples, which is going to track which samples belong to which groups. Here we've got 4 days that these samples were collected from tomatoes. And then within each of the biomolecules, you'll also have a third file that tracks any additional information, which I added classes, and then the statistics, which has all the full changes in p-values. Now, to identify trends across these omics data sets, you can use trelliscope displays, which are very interactive and highly shareable because they're HTMLs. But to actually make one, it requires quite a bit of coding expertise. So we made an app. So after you upload your data from which Rachel showed you, we've got 3 ways that you can group the data by either the biomolecule class, the samples, or by the biomolecules themselves. We support 3 types of plotting right now for abundances, full changes, and missingness for a total of 8 plots overall. After you select which kind of plot you want to make, you can explore some examples, confirm your selection, and then modify it. And this kind of removes all of the expertise in learning how to use ggplot packages and everything else. You can just make a nice plot quickly. Here I'm renaming the x-axis to something besides group, days, because that's more useful for my users. I'm changing the y-axis to just normalized abundance. And then I make the text bigger because I am going blind. So after you make your modifications, you can change the name of your trelliscope display and then pick cognostics, which are these nice little labels, 1 per plot that you can add for information to sort your data by or to explore trends. So once you have your finished HTML display, this is something that you can easily use and share. And so at this next step here, I'm going to actually show you these displays. These require no coding expertise. You literally just drop an HTML file in a folder to someone else. They can open it and use it. It's been highly successful on multiple projects for sharing this kind of information. Here we're looking at the full changes. We're looking at the normalized abundances for each of the days across all these biomolecules. You can set up grids for which kinds of plots you want to look or the way you would like to order it. You can add labels, which is going to happen in a second. It's also very easy to click through these plots. So here I'm interested in biomolecule class information that I just added myself into the file, and then I was able to pull into the trelliscope display. And then here I'm interested in looking at amino acids. And so this kind of allows us to see trends across all the amino acids for this data set. For these tomatoes, we see an increased expression from day 8 to day 55. For most of the amino acids, it's the acceptance, with the exception of tryptophan here. And so this would be really difficult to look for in just an Excel sheet or by making lots of plots and hoping you find something. Here, we're kind of removing that extra time needed to identify these trends. You can also add other plots. So I have this bar plot for the direction of the full change with an outline if it's significant. So here with alanine, it doesn't look like there's a significant change just visually, but if you add the bar plot and the results of our statistics from PMARD, you can actually see that is a significant change. I'm also interested in that tryptophan example now where we see that decreased expression. Is that also significant? And so that's the next thing I'd look at. And then there was one other between day 8 and day 55, which is the comparison of this bar plot here, where I wasn't sure if there was a significant change, and there isn't. So this is just a really nice way over like 4 minutes to quickly show that we identified a trend with amino acids in this data set. Out of 11 or 12, we see significant change. 10 of those is an increased expression, 1 is a decreased expression. This is a nice thing to show your PI on a project, and then you can go from there and answer the questions as to why you saw that. So thank you for your time. I have a nice little poster, so go see it. Thanks.
Thank you, David. Great. All right, so we're going now. Our next speaker is Jeremy Jacobson. He's a master intern here at PNNL. Please, Jeremy.
Can you all hear me all right? Sounds like it. So I'm going to be presenting on the Mobility dashboard today. This is an application designed to analyze mobility mass spectrometry data. And I'm just going to start off with figure 1 up on the left. And so this is going to kind of highlight the usage of why this is important in the first place. And so we've got mass spectrum. This is MS data of structural isomers and enantiomers. And so between glucose and fructose, we can see that all these peaks are in the same locations. And so with MS data, with all these peaks in the same location, unless you have really clean data, it's going to be very difficult, and really good reference data. It's going to be really difficult to tell these structural isomers apart. This problem is even more difficult to tell apart with enantiomers. With L-lactic acid and D-lactic acid, there is essentially no difference between those two plots. And so that brings in ion mobility mass spectrometry data. And as mentioned earlier today, this can help provide the difference that's needed between the structural isomers and some enantiomers. And so we had a great graphic earlier today that demonstrated how ion mobility mass spectrometry data works. And that's, you have this long drift tube filled with the buffer gas. As you propel your ions through the gas, it collides with these and they separate out essentially by shape and size. And so this orthogonal dimension is what's used to tell the difference between the structural isomers. And there's one main difficulty with this data, which is the analysis portion. And so currently, if we're looking down at figure 2, these are each of the steps that are required to analyze this. And so in the red, we've got tool names, starting off with PNL preprocessor through ProteoWizard, then MZMine or DMOS, and then ending with Auto CCS. In the green, each of these green tabs are intermediate scripts or steps that are required as well. And so quickly, just running all these tools with these intermediate scripts becomes difficult to process this data. And so this is what we're trying to solve in this application. And so looking up at figure 3, we've got a screenshot of the ion mobility dashboard. This application essentially joins together all of these tools and intermediate scripts so then users can essentially select the files that they need, enter parameters, and then hit run, and it'll run it through the pipeline. If you looking at figure 3, we have workflow on the far left of it. And so under that tab, there's single tools, so you can run these tools individually, or there's also several pre-configured workflows. These are for single field, step field, and Slim. And Slim was generated or is produced here at PNL. And so with each of these pre-configured workflows, all you have to do is select it, enter in the files, your file paths, which you can just browse and select. And then you run it going through, you just hit run, and then looking at figure 4, it sends that into pipeline.py. And what that does is it's essentially an orchestrator script for these Docker containers and these Docker imagers that we have below. And for anyone who's not familiar with Docker, this is essentially just an environment that'll run the same on different systems. So this will run the same on Mac and Windows and on different computers. And so this allows these to all run together. And what pipeline.py does is it just calls each of these steps. And once these steps are completed, it returns the results back to the ion mobility dashboard, where you can view results or save them or run a new experiment. And if you have any more questions, I'll be at poster session 1.